of Glasgow in the UK, who's going to speak to us on telltale signs of a multi-species epizootic spatial epidemiology of HPAI during the 2022 seabird outbreak.
I think a curse today.
Yeah.
Okay, thank you very much for staying this late. I've never attended this conference before. It's a real education for me. I've looked to listen to particular messages from the conference, and at least two of the messages that are recurring are, first of all, how important wildlife is for the whole One Health problem that we're facing. It's great to have. So, yeah, and it's great to have half the conference congregated in this tiny room. And the second big message is just how difficult it is to make any sense of the very biased, bitty, gappy, imperfect data that we have at our disposal. So this talk is nominally about Gannets, but actually it's about reconstructing inferences from such imperfect data. And again, it's nominally about Gannets, but it's actually about what kind of inferences can we draw from one species about the whole seabird community in this case. So essentially, I'm talking about torturing data to an extreme until they spit out some answers. Now, we've been working on the Gannet metapopulation system in our group for more than a decade, and actually standing on the shoulders of giants by kind of analyzing data that are a century long for the entire sort of European metapopulation system, and Jana gave a really good overview of that. But even though we sort of been working on the whole perspective of dynamics, spatial distribution, movement, and epidemiology, we were certainly caught unprepared when the really radical, drastic outbreak struck in Gannets in our study species. Took out about a third of the Gannet metapopulation, and it burned really fast, really, really badly through the entire system. So we were tasked with trying to understand what happened by using very intermittent, opportunistic, partial data. Stephanie's talk really sort of covered the difficulties of partial observation. And the sort of two questions that we want to try and address are, first of all, why? And second, how can we ever do this with data being so biased and imperfect? Now, we have some mechanistic insights, and the conference has given us some more mechanistic insights, for example, to do with movement. And Jana mentioned those results. I'll just put them up. So even though in the long term these birds seem to be unaffected in their movement patterns by the onset of HPAI, in the short term, right where it matters, where they're affected and infected, they seem to do uncharacteristically long movements. Which kind of opens up the possibility that HPAI in Gannets is actually a self-propelling, self-propagating disease. It kind of triggers its own dispersal, which is an interesting prospect. I think my sort of somewhat arrogant conclusion is that it's going to be really difficult for us to piece together a large picture of what's going on in the wild by building on such incremental mechanistic insights. I think we should be collecting them, we should be moving towards them, but I think we also need a top-down kind of analysis of the patterns that we see in the body counts, the carcasses. And this is what we did. So the core of the modeling work that was involved here is not really profound. It was a compartmental model, such as the ones that you see in epidemiology all the time. But the key differences between this and most of the models that you might have seen are two. First of all, this model operated on all 46 colonies, assuming connectivities of different types between them. So it was a metapopulation epidemiological model. And the second difference is that it was not just parameterized and run, it was fitted to carcass data. So it was fitted statistically to carcass data. And the sort of results that you get when you do something like that are, at a minimum, a reconstruction of the kind of carcass counts that you observed in your data, and a reconstruction of the epidemic as it played out in the entire metapopulation, and the final outcomes in terms of deaths and recovers and so on and so forth. But actually, you can get specific inferences about particular parameters of interest that are really usually non-observable in the wild, such as the basic reproduction number, which in our case was estimated very precisely to be quite a bit higher than what's been reported in the literature before, and also things like the carcass detectability probability, which relates to Stephanie's talk again, which in our case proved to be about barely 10% of the dead bodies that the model estimated to have died. But I think the connectivity aspects, the linkage aspects between the colonies are the ones where a lot of insights can be had. The model contained two types of links. The first one was the connectivity in the sense that you would understand it in an everyday context, which says one gannet colony has a certain capability to give HPAI to another gannet colony depending on how far away they are. And you can think about this as a network. So we modeled that connectivity. And when I say we modeled that connectivity, the parameters of that connectivity were estimated at the same time as the epidemiological parameters of what was going on within the colony by fitting the model to the carcass data. So everything that I'm showing you is being inferred simultaneously. This is not a piecewise kind of estimation process. But the second kind of linkage is a little bit harder to kind of intuit. What it did was it used the gannet colonies as a network of observation points to say, is there over and above the patterns of connectivity that we see on the left, any residual patterns in space that could be the result of the disease moving around spatially due to things other than gannets, other species? So that's the sort of synchrony mechanism. Both of these mechanisms were embedded in the model, and we tried to see whether the model could tease those apart, essentially the within species and the between species transmission. And what are the scales, the spatial scales of that type of transmission at large? Magically, it turned out that these imperfect data were able to not just give us the epidemiological parameters for HPAI, for the gannets, but also to tease apart these spatial mechanisms. And what I mean by that is the sort of picture you can get is the spatial scale over which these mechanisms are operating. So in blue, what you see is the capability of gannet colonies to pass the disease to each other. And you see that the half distance is tiny. It's about 5 kilometers. In contrast, the synchrony mechanism has a half distance of about 98 kilometers, so it operates at a much farther ranging range. Now, the median distance between gannet colonies is about 94 kilometers. So it takes several large colonies to be undergoing a very severe outbreak in order for the blue mechanism to actually become operational, in order for that really thin tail on the right to generate some cases in faraway colonies. Now, by having those two mechanisms and having inferred those two mechanisms, what we were able to do was to reconstruct a time series of who was responsible for the gannet outbreak. Was it the gannets or was it everybody else? And what you see there is that in green, we have everybody else. In blue, we have the gannets. And essentially, the story is that everybody else was driving the gannets to begin with until they took over and sustained the disease themselves. But more to the point, we can reconstruct the story spatially. So we can essentially reverse engineer this other species mechanism to get an idea of how the disease was sloshing around at the sea around Europe during that period. And this is essentially what the picture looked like. So this is our best guess, just looking at the gannets, of how the multi-species community was experiencing HPAI through the entire outbreak at the time. And that's without having any other species data, just using the gannets as essentially a sentinel species. So to conclude, these data can be pushed actually quite far. It seems that network connectivity and background infections interplayed to generate this vast outbreak for gannets. It seems that HPAI strains, whatever they were more virulent than those described in the literature. And key recommendations or wishes from me are the two points at the bottom. I would very much like to see this kind of epidemiological approach come together with a lot of the phylogenetic genetic approaches that we've been looking at for the last couple of days to see how we can approach this problem from both ends. And I have some ideas about that. And the second one is that perhaps as a community, we might develop frameworks such as this into early warning observation or sentinel systems, where we may be seeing very small bits of what's going on, but we can reconstruct actually some quite complete dynamic pictures of the things that we can't really see. Thank you very much.
Thank you very much, Jason. That's a complicated story, very well told. We have a couple of questions here. Well, what about contact or contamination other than colonies?
I'm not sure I understand. I'm not sure I understand the question either than between colonies or than within colonies. So I'll probably say again what I said in the talk. There were three epidemiological processes that the model was looking at. It was looking at the within colony epidemiology. So once the colony caught the disease, how did it spike? It was looking at the between colonies in the Gannet species and external drivers from things other than those two. I can't understand the question any further to give a better answer, I think.
Okay.
If the person's in the room, by all means.
Any clarification wanted from the floor? You happy? Okay. We'll go in with another question. Where did the carcass counts come from? Have you tried rerunning results, assuming that breeders missing in 2023 had died in 2022?
Assuming that the breeders missing in 2023 have died. So the model did contain some sort of additional data. So it wasn't just using carcass data. Admittedly, it wasn't using future years of missing breeders, but it was using estimates of overall mortality at the end of the outbreak for selected colonies where those were available. It was also using information on black eyes to give information to the model about exposure for those colonies where those were available. So in principle, the incorporation of very different types of data is fundamentally possible, and I think it's a really good suggestion to bring sort of future years in as a post hoc corroboration of what was happening. It's a fundamentally great idea. Where the data came from, it was a diversity of sources, and quite a lot of effort went, not by me, admittedly, into collating those. Yeah.
Thank you very much. And we've got one last question from online from Thijs Kuken. Did you take species differences in susceptibility to HPAI into account in the model?
There was only one species in the model.
Okay.
So the rest of it was just essentially modeling the residual signal of external drivers in the force of infection.
Okay. Thank you very much, Jason. Thank you.